Let's continue with another video. In this video, we are going to talk about recombinant DNA. Molecules are DNA molecules formed by the laboratory method of genetic recombination, such as molecular cloning that bring together genetic material from multiple sources, creating sequences that would not otherwise be found in a genome. Recombinant DNA is the general name of the piece of DNA that has been created by combining two or more fragments from different sources. Recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure, offering only a nucleotide sequence. Recombinant DNA molecules are sometimes called chimeric DNA because they are made of material of two different species like mythical chimera DNA technology used with palindrome sequence and lead to the production of sticky and blunt end. A DNA sequence has to be construction of recombinant DNA. Molecules can be originate from any species. For example, plant DNA can be joined with bacterial DNA or human DNA can be joined with fungal DNA. In addition, DNA sequences that do not occur anywhere in nature can be created via the chemical synthesis of DNA incorporated with the recombinant DNA molecules. Using recombinant DNA technology and synthetic DNA, any sequence can be created and introduced in living organism. Protein can be result from expression of recombinant DNA with living cells are termed recombinant protein. With recombinant DNA and encoding a protein and introduced in a host organism. Recombinant protein is necessary to produce. Expression of foreign protein require a use of sequenced expression vector and after necessity is significant restructuring of foreign coding sequence. Recombinant DNA differ from genetic recombination in that a former result from artificial method while later the normal biological process that result in a remixing of the existing DNA sequence in essentially all organisms. Molecular cloning and laboratory process used to create a recombinant DNA. It is one of the most widely used method along the polymers chain reaction used to direct and replication of any specific DNA sequence with chosen by the experimentalist. There are two fundamental differences between the method. One of the molecular cloning involve replication of DNA with living cell or PCR replicate DNA in the test tube. Free of living cell, the most difference is that cloning involve cutting and pasting DNA sequence by PCR amplifies the copying existing sequence. Formation of recombinant DNA requires a cloning vector or DNA that replicate within the living shell. Vector are generally derived from plasmids or viruses are represent a relatively small segment of DNA that contain necessary genetic signal of replication as well as the additional element of convenience in inserting foreign DNA identification shell that contain recombinant DNA or where appropriate expressing the foreign DNA. The choice of vector of molecular cloning depend on the choice of the host organism. The size of the DNA to be cloned, whether the used to the foreign DNA to express the DNA segment can be combined by the variety of methods such as restriction, enzyme level cloning of Gibson assembly. In a standard of cloning protocol, the cloning of DNA fragment eventually involve 7 steps: choice of host organism, cloning vector, preparation of vector DNA, preparation of DNA to cloned, creation of recombinant DNA, introduction of recombinant DNA to win a host organism, selection of organism, selection recombinant DNA, screening of clone or desired DNA insert, and biological properties. These steps are described in some detail and related. DNA expression are required to be transfection of suitable host cell, typically and by either by bacterial, yeast, or insect of mammalism cells are used to host cell. Following transplantation of into the host organism, foreign DNA contained within the recombinant DNA construct may be have expressed. This is the DNA simply replicated without expression or maybe transcribed and translated in a recombinant protein or produced. Generally speaking, expression of a foreign gene require restricting restructuring or to genetically sequence that are required of producing mRNA molecules that can be used to host transplant apertures. Example: promoter, translator, initiation signal, or transcriptional transmitter. Specific change in the host organism. may be made to improve expression of ectopoic gene. In addition, changes may be named needed to coding sequence as well to optimize translation, maybe protein soluble direct to recombinant protein to be proper cellular extracellular location and stabilize the protein from degradation. Properties of the organism containing recombinant DNA. In most cases, organism containing recombinant has a apparently normal phenotypes. This is their appearance, behavior, and metabolism are usually unchanged, and the only way to demonstrate the presence of recombinant sequences examine the DNA itself, typically used to polymerase chain reaction. Significant expectation exists and discussed below. If the rDNA sequence are encode a gene that is expressed, then the presence of RNA or protein product are recombinant DNA that can be detected, typically using RT-PCR Western hybridization method. Gross phenotypic changes in not norm unless the recombinant gene has been chosen or modified in the gene biological activity in the host organism. Additional phenotype are encountered include toxicity of host organism induced by the recombinant gene product, especially if the overexpressed or expressed within the inappropriate cells or tissue. In some cases, recombinant DNA can be deleterious effect even if the not expressed. One of the mechanism which has happened in certain inactivation in which rDNA become the inserted into host cell and gene. In some cases, researchers use the phenomena of knockout gene to determine their biological function and importance. Another mechanism to which rDNA insertion into a chromosomal DNA can affect gene expression by inappropriate activation of previously unexpressed host cell gene. This can happen, for example, which is recombinant DNA fragment containing an active promoter become located next to previously silent host cell gene. When the host cell gene has a function of restraint, gene expression undergoes insertion activation by recombinant DNA. Application of recombinant DNA. Recombinant DNA is widely used by biotechnology, medicine, or research. Today, recombinant protein and other products have result from use of DNA technology found in essentially every Western pharmacy, physician, and veterinarian office, medical testing laboratory, biological research laboratory. And in addition, organisms that have been manipulated using recombinant DNA technology as with product derived from the other organism have been their own. Many farms, supermarket have a mall medicine cabinet, and even pet shop, such as those shall glowfish or genetically modify animal. The most common application of recombinant DNA is basic research, in which the technology is important for most current work in biological and biomedical science. Recombinant DNA is used to identify, map, and sequence genes to determine their function. DNA probes the employed analyze the expression with individual cell throughout the tissue of all organisms. Recombinant protein are widely used in reagent and laboratory experiment and generate antibody probe to examine protein synthesis within the cell of an organism. Many additional practical application of recombinant DNA are found in industry, food production, human and veterinary medicine, agriculture, and bioengineering. Some specific examples are identified below. Recombinant cynomosine, cynomosine, mosine, cynomosine, found in the rennet. Cynomosine is the enzyme required to manufacture cheese. It is the first genetically engineered, both additive and commercially traditionally processed obtained cynomosine were from rennet in a preparation delivered from the fourth stomach of the milk feed calves. Scientists engineered a known pathogenic strain E. coli bacteria for a large scale laboratory production in enzyme. The microbiology produced recombinant enzyme identical structure to cost less than the produce of the adaptant qualities. Today has been 60% of the US has been cheese is made with the genetically engineered cynomosine. In 1919, FDA granted cynomosine generally recognized for safe studies based on the data showing of enzyme was safe. Recombinant human insulin almost completely replaced insulin obtained from animal sources like pigs and cattle are treatment for insulin dependent diabetes. A variety of different recombinant insulins preparation are widespread. Recombinant insulins are synthesized by inserting the human insulin gene into coil yeast which has been produced insulin for human use. Insulin produced of E. coli requires further post transit modification, where yeast are able to perform themselves by virtue of being more complex host organism. The advantage of recombinant human insulin is that chronic use of patient don't develop immune defense against if the animal source insulin restimulates the human immune system. Recombinant human growth hormone administered to patient whose pituitary glands generate insufficient quantities to support normal growth and development. Before recombinant HGH become available, HGH is therapeutic used by often for pituitary gland for cadavers. The unsafe practice lead to be some of the patient developing recombinant blood clotting factor 8, recombinant hepatitis B vaccine, recombinant antibodies, diagnosis with infection HIV. These are the three widely used diagnosing HIV. Has been developed using recombinant DNA. The antibody test uses recombinant HIV has to be test presence of antibody that has a body of produced in response to HIV infection. The DNA test look for the presence of HIV genetic material has reverse transcription polymers chain reaction. Development of the RT-PCR test has been made possible by the molecular cloning and sequence analysis HIV genome. HIV testing has been a US Center of Disease Control CDC. Golden rice, recombinant varieties of rice that has been engineered to express the enzyme responsible for V cartoons biosynthesis. The variety of rice hold substantial promise to reducing incidence of vitamin A deficiency in world population. The gold rice is not currently in use, pending the resolution of regulatory and intellectual property. Do you get about it? Harvick resistant crop, insect resistance crop. Vaculus is a bacteria. Bacterium and a naturally product as a protein with insectual properties. The bacterium has been applied to the crops of insect control strategy for many years. Practice has been widely adopted in agriculture and gardening. Recently, the plant has been developed and expressed in a recombinant from the bacterial protein, which has been effectively controlled some insect predator aromatic issue associated with the use of the transgenic crops has been fully resolved. History. The idea of recombinant DNA has been first proposed by the Peter Levin, a graduate student of Professor Del Kaiser of the Biochemistry Department of Stanford University Medical. The first publication describing a successful production of the intercellular replication of recombinant DNA has been up in 1972 to 1973. That's from UCF 1980 Paul Work and a professor of biochemistry department of Stanford and author of the first paper as awarded Nobel Prize of Chemistry for his work on nucleic acid. With particular regard to recombinant, winner award Hamilton. Stanford University appeared to the US patent in recombinant DNA in 1971, listing the investor Herbert V. Boyer, professor of University of California, San Francisco. Stanley and Cohen, professor of Stanford, their patent was awarded in 1980. The first licensed drug generated using recombinant DNA technology was human insulin, developed by Genentech and licensed by Eli Lilly and Company. Controversy. Scientists associated with the initial development of recombinant DNA method recognized that potential existed organism containing recombinant DNA to have undesirable and dangerous properties. At the 1960s Assembly Conference and recombinant DNA, these concerns were discussed on virtually marathon, and recombinant DNA research has been initiated experiment that has been considered particularly risky. The moratorium widely observed that a National Institute of Health developed issued for formal guidance of RDNA today recombinant DNA. Recombinant protein are usually not regarded as dangerous. However, concern remain about some organism that spreading the DNA, particular when they leave the laboratory or introduced into the environment food chain. These concerns that discussed in article genetically modified organism and genetically modified food controversies. Furthermore, there are concern about the byproduct and biopharmaceutical production where recombinant DNA result in specific protein products. The major byproduct termed host cell protein come from the host expression system and poses a threat to the patient health and overall environment. You get it? What they want to set this through article. That's it for the video. We'll see you next time. Already 15 minutes has been covered in this video. Do you get it what I want to say? Already 15 minutes has been passed in the video. So let's stop it.